Hey and welcome. Today I've got Sven with me. He studies with me neuroengineering and he's a smart guy. That's why I got him here to talk about a very important topic, and that is whether you should get into science. Because we are at this crossroads where you decide to either get a job, become an adult, or continue to stay a student for life and become a PhD and
Stay a slave,
stay a slave.
yeah.
Right. As you may notice, there is already a sort of pessimism in this room
Slight, yeah.
concerning that topic, and we would like to really focus on all of the points today that we'd like to address about what the problem is with science nowadays and why it is wise in a lot of cases not to pursue it. And I would say let's get straight into it. We've got the very famous example of the replication crisis.
Mhm.
Explain to us what the replication crisis is, please.
Yeah. So I'm a bit pessimistic about doing a PhD and one of the big reasons is the replication crisis. I'm not saying that science itself is a bad thing. It's actually a good thing, and it's something that should keep society driving.
Right,
However, then there's a replication crisis.
right,
And the replication crisis essentially means that lots and lots of the studies, and in most fields, this is more than half of all the studies that are published, they are
right,
not replicable. So that means when other researchers try to replicate the same results and follow the methods to be exactly the same, they don't come to the same conclusion. Yeah.
right. And how was it assessed that they're not replicable?
Well, you can take the findings. Let's say you take any journal
Right,
and you have something that is statistically significant, which I
right.
think we will probably talk about.
Yes,
Then another group comes along, they do the same procedures, and suddenly they realize, okay, for us, it's not significant. And
right.
then, well, the question is, why is that the case?
Mm. That does relate to what we're going to talk about, p hacking as well, right? So you can
Mm-hmm.
have you do the same experiment twice, and then at some point you're
Mm-hmm.
at below 0.05, and then you consider it your result, right?
Yeah, exactly.
I see. And would you say there's some fields that are more replicable and others that aren't? Is this something that's like maybe not um,
Mm.
general, generalizable? Is there a replication crisis in some fields and in others there isn't?
Um, okay, this is like a sub-educated guess I would make.
Right,
It's probably for those fields where you can have lots of um, third factors playing into your studies. For instance, psychology.
right,
If you would like to study something like attention, then it might also play a role what the lighting in the room is, or what the
right,
temperature in the room is, etc., etc. And if you don't account for these factors, then you're very likely to miss something that actually affects what you're studying.
right. I see. I could imagine, for example, some fields like physics, experimental physics, where
Uh-huh.
they shoot electrons in a vacuum chamber against some material. I can imagine that being quite replicable because you just have to have the same setup.
Yeah,
Um,
I remember that there was also like I think a smaller crisis also for physics
okay,
to some
okay,
extent.
interesting.
Yeah, to be honest, I'm not 100% sure. I just read on it
Right,
on the side, but I know that usually what is often done in physics is the p-value, which is kind of the level after which you assume something is not coming from the same distribution,
right,
i.e., it's significantly different,
right.
is usually a bit lower because I think they use 0.01 or 0.001
Right.
even
Okay.
for a lot of things. So that also makes a difference. Yeah.
Right. So it's kind of harder to pass the test in a sense.
Yeah. Or to
Right.
find something that actually, yeah, is significantly different.
Right. Well, the way to understand it, maybe for the audience, is that if you have a distribution of effects and the null hypothesis is there's no effect, then you can assume that your hypothesis is true if if it had been sampled from the original distribution, that probability would be very low and oftentimes
Yeah.
below 0.05 or below 5% in that
So,
sense.
I mean, here the term population is not like the general um, term inside the population. So we're not talking
Right.
about population of a country, but population could be, for instance, a control group
Yeah.
that didn't receive a treatment versus a group that received the treatment.
Right.
And usually what we do is we take samples, because we cannot test all of the people in the population because often it's too big. Right?
Yeah.
So we take a sample and we want to compare the two, let's say the two means of the groups, because we want to know if we apply a treatment, if the treatment is effective.
Yeah.
Right. And we want to see if when they had the treatment versus not, whether the mean is going to differ in a positive sense.
Yes.
And so we assume under the null hypothesis, you would say we have a distribution, which is like usually this bell curve, right? This is a bit hard to show with my hands. And under that distribution, we kind of ask, okay, what if a sample is extreme enough, or when would a sample be extreme enough such that we consider it to be from a different distribution? That's essentially what we try to do. And then because you have this bell-shaped distribution, you can say, well, you can take the mean and the distance from the mean as sort of an indication of how unlikely something is to occur if it were from that population.
Right. Yeah. That makes sense. A general question about statistics there is like, isn't already modeling all these results via bell curves, for example, an extreme like reduction in a sense, right? You kind of putting them all in the baskets, putting this like curve over it, and then if you even then make mistakes, or even then kind of become very sloppy in your method, then there's really no rigor left to your procedure, right?
Yeah, I mean the reason we usually use the bell curve or this Gaussian distribution is actually the central limit theorem, which essentially states that if we have a population with a specific mean and a certain variance, then if we take multiple samples, and it's called a sampling distribution, and we calculate the mean for each sample, that the distribution of those sample means will approximately be normal as the sample size increases. That's what it says. And it will have the same mean as the population what we sample from, and a variance that will be divided by the sample size, which is kind of the factor that makes sure that for small sample sizes you will have like a large variance.
Right.
Which also is a problem with a lot of studies with small sample sizes, because we only take one sample. We use the estimate to, for instance, calculate a t-test.
Right.
And depending, on that, you might have a large variance. And I mean, generally it makes sense. Imagine you have a large population, you take a very small sample. Let's say you want to know what the size of the population in Germany is.
Right.
And you take two people and then you take the mean of them. How valid is that estimate of the actual size in the population?
Right. And then you'd say that to be statistically rigorous in your method, you'd have to assume a massive variance if you have a sample size of 2. How would you arrive at what variance you'd have to use?
In that case, I mean, with 2 people,
but let's assume you're being methodically sound and you have only 2 people. What variance would you have to use to be fair? You know what I mean?
I mean, according to the central limit theorem, you would compute the variance and divide by the sample size. But I don't know if anyone should ever do, like, if you do a study with 2 people, which is usually case studies, you should probably not use inferential statistics, but describe what you see. I'm not saying that it doesn't make sense. Sometimes when you want to try out some exoskeleton or something, just build it and test it on one person, and just see if it works. Fine, but don't do hypothesis tests with it.
Right. How would that sense of variance tie into if you have a too high variance now in machine learning, for example? Because you also have that problem of a lack of generalizability, right? You have a lack.
Yeah, I mean, this is the essential difference between what I would call explanatory statistics or explanatory modeling, where you take a sample and you make a model. For instance, let's say just a simple linear regression, and you try to explain the data that you have as good as you can, right? And if you imagine you have 20 different data points and you make a regression with with many parameters, such that you can get like a perfect line that crosses through each of the points, right? It would perfectly explain your data set.
Right.
The problem is, if you would take another data set, it would very likely not generalize.
Right.
And that's the difference to predictive modeling, i.e. machine learning, where it essentially only matters how good you can predict new data that has not been in the model fitting before.
Right.
Yeah.
And that is also an issue, that a lot of studies that should be able to be predictive in order to be seen as a good theory or a good hypothesis just work with explainable statistics. And that is a kind of lack in the quality of that research. It's something that I've heard you at least criticize.
Yeah. I mean, what people should not mistake is the p-value is not an indication of how predictive something is, right? Because, as I said, you fit a model to your data, and that model hopefully explains your data really well, but it does not explain unforeseen data very well.
Right.
And if you have a very small sample, then I don't know, any model you can do from a small sample, and people that do predictive modeling quite often, they know, like, when you train from a small sample, it's usually better. The more data, the better. As
Well,
long as
let's
you can
stick to the
handle it,
example with the height of the population of Germany and take a sample size of 2. That's the problem we have, right? But like in that case, how does that word variance fit in this case? Because like you say, you can compute the variance in that sample set of 2 people, right? But that wouldn't have to be a very high variance. Why do you call it? Why would you say then in the case of machine learning or predictive coding that is a model with high variance?
Well, in the case of predictive modeling, you have the so-called variance bias trade-off, where high variance would be you let's say you make a model and that perfectly explains your data, the sample that you have.
Right.
But then you try to use that and make predictions to data that you haven't seen.
Yeah.
And then your predictions to other data sets, let's say you have several of them, there will be a high variance in how well you can predict it, or what the accuracy, for instance, would be. Whereas the bias, something with high bias, would be a model that is that essentially sucks at explaining your data.
Right.
And you basically try to get like the trade-off of both. You want something that explains your data at hand quite well. Let's say your training set. You don't want a model that has like a huge error on your training set.
Right.
However, it's also not good if you have a very small error on your training set, but the error on your validation set is small.
It's massive. Yeah.
Yeah.
Right. I see. Now that's one thing. That's a personal thing. Now, one thing I see in a lot of studies is, as you mentioned, a linear regression or something. Just as an explanation, in the basic way you get it in a lot of studies, that you have data points and you try to have some sort of explanation or some sort of description of the distribution by putting a line through the data set, right? Simple scenario. And you could say in computational methods, it's more higher dimensional. Can't necessarily put it in a graph on a paper, but like I see a lot of graphs, and often there's just a cloud of points scattered in it, and then there's a line drawn in it, right? A line drawn in it. So I know that like if you do the proper calculations in a lot of ways, in a lot of cases, there is a good reason to put the line in there like that. But is it something that you've noticed as well, that it's sometimes, a very, a very courageous kind of line that's drawn into this cloud of data points?
Yeah, I mean, it would be an example of high bias if you would put, like, let's say you try to describe a certain shape of data with just the mean. That would, for instance, be like a very high bias model. I mean, generally, I would criticize that lots of people try to do hypothesis testing, and then they use like 2-3 subjects and they draw like a strange line, and it's very likely that if you take more patients or subjects, whatever, that this line will look very different, because you have a very, very small subset of the actual population, and you should not attempt to model the population with such a subset.
Right.
Yeah. But even if you do everything correctly and you have large sample sizes, that it's still the explanatory models would underestimate the actual error.
All right. And I think this is a good point to get to our main part or final part of this, and that is the findings of John Ionnidis about how most of published research is false. And that is quite a claim, right? But that is kind of the climax of what we're talking about. And so how does it happen? How can it happen that you have so many scientists that are well trained, but yet you come to a result that, you know, most of the research is false? Do you think Ionnidis is like a pessimist? He just wants headlines, or what is the background of that? And like, is it even solvable?
Yeah, I mean, one thing is what we discussed before, the nature of explanatory modeling itself, or fitting to a sample. The other thing is that when we make this arbitrary decision of 0.05, right, which is like the p-value upon which or under which we say something is significantly different. When we do that, which is really arbitrary, it was just suggested by Fisher in something in the beginning of the 20th century. It means that we kind of accept in 5% of the cases to reject the null hypothesis, even though it was right. We shouldn't have rejected it. And
the null hypothesis being that there's no difference.
There's no difference.
There's no
exactly.
difference.
Yeah. So it would be a false positive.
Right.
Yeah. However, that's a drastic underestimation. At least that's what Ionnidis says, and I think he has a point there. I mean, the question is how drastic that is, and that's hard to answer. However, what Ionnidis says, and it's kind of similar to this medical test paradox, he says we essentially don't know the prior. We don't know, let's say, over 1,000 hypotheses that researchers make in a certain field. Let's say that 10% of them are investigating an actual effect that is there. Right. That will mean.
But this is like a guess, right?
Yeah, this is just a guess. It's hard to estimate that.
It's hard to estimate. Yeah.
And then let's say we have a certain statistical power, and statistical power means that statistical power is the probability that we find an effect if it's really there, right? And that also, again, depends on the sample size. The larger the sample size, the more power we have, and you can also pre-calculate that, etc.
Right.
But let's just take that for granted now, statistical power. And let's say it's around 80%. That's usually a value that a lot of researchers try to shoot for when they do the studies.
Right. And you shoot for it by increasing the sample size.
Yeah, you can basically pre-calculate what sample size you would roughly need to achieve that. And then let's say, as I said, we have 1,000 hypotheses. 100 of them investigate an actual effect. That would mean with a power of 80% that we would get 80 of the 100 actual effects and find them.
Right.
Right. We would see a significant difference, and that would be true. However, that also means of the other 900 hypotheses that we would have 45 false positives, right?
Okay.
That means in total we would have 80 plus 45.
How did you arrive at 45? That's 5%.
Yeah, 5% of 900. Sorry.
5% of 900. Yeah. And that's because you chose the p-value of 5%.
Yeah, exactly. We accept that we do that mistake in 5% of the cases.
Right. That means 5%.
Or we have a 5% probability of making that mistake. So.
Right. So we have a 5% probability of having a false
positive.
Yeah, false positive out of all of these false hypotheses, right?
Yeah.
As
so
well
essentially we assume an effect even though there is none.
Right.
And that would mean that we have, from the 100 hypotheses that actually investigate an effect, as I said, we have a power of 80%. We get 80 of them right. However, we also have 45 positives of the 900 other hypotheses. And that would mean, I don't know, you can do the math. 80 divided by 80 plus 45. That would be
plus 20. What about the 20 that we have?
no, this is just the rate of things that we see. Okay, what was deemed to be a significant effect, and how much of that is an actual effect. That's what it would calculate. So it would be 80 divided by 125.
Ah, I see.
Yeah.
That means you'd have a significantly lower percentage of papers just by applying the statistics
because
correctly.
Of the prior.
That would be right.
Because of the prior, and that doesn't even and that's kind of the point that he also makes in this paper back then, is that this is if you do everything correctly, right?
Right.
This doesn't include stuff like p-hacking. It doesn't include people that really just fake their papers.
Right. Just for information, p-hacking is more or less you repeat the experiment until you have reached a p-value below 5%, and thus a form of cheating in a sense.
Yeah, I mean, as we said, you have like this 5% probability of making a mistake, and the more often you do something, eventually you will get to an event where that mistakes happen, right?
Yeah. And I can do roll dice, and I can make a study about how dice you always roll a 6. I just need to do the experiment often enough, and I'll have a row of 100 6s.
Yeah. So don't do that.
Don't do that. It's bad. It's wrong. So okay, and that's and the interesting part here is that it shows that even if you do it correctly, that would render a lot of them false. In Ionnidis' study, depending on the prior, he arrived at most, like the title says, most, it was over 50% that are false. The problem is it really depends on the prior. It really depends on what we assume, or we can assume how many of our hypotheses are correct. I think it's realistic to assume that the minority are correct. Otherwise, science would be super easy.
But I mean, like, I really want to point out there's like super many other things that play into, right? It's also and I think that's why some students also wouldn't want to pursue a PhD. It's not just the great money you earn there, but it's also the fact that a lot of researchers are kind of forced to publish, to sell themselves in that system.
Right.
And this actually, and also the publication bias, meaning that negative findings are not as likely to be published as positive findings.
Right.
That just increases that problem
a
lot, right? So it's actually a real crisis, I would say.
It's a real crisis of science where you really don't know whether you can trust a particular research publication, and that pressure that you have also really kills the kind of idealistic scientific drive. So that really makes you think twice over pursuing such a career, and that is essentially what we're talking about. So we've got especially a structural issue. We've got a whole institutional issue, you could say. You get these peer reviewers that are not paid, that are just part of the whole system that needs to just publish to have your credits, and you don't get paid well anyways as a scientist. And so it's also becoming competitive when you look at the amount of people going to university, graduating in university, and the amount of limited PhD positions, right?
Yeah.
So that's a real problem you have, and it doesn't really contribute to the good image that science has. And I feel like there's a lot of dishonesty because they want to maintain the good image. It's like they want to have their cake and eat it too. They want to maintain the good image whilst not internally maintaining it.
Yeah. And I mean, what I'm not trying to say is that science itself is bad, right? I mean, if science is done correctly, it's probably the thing that should guide our society. So I do love science. However, remaining idealistic and then going into the PhD and knowing you have to do all of this, and you kind of sometimes even almost maybe forced to engage in, even if you do it correctly, forced to publish something where you're not sure if that's actually helping in increasing awareness about a certain phenomenon. Do you really want to do it if you're an idealist?
Right. So I hope this discussion has opened some eyes and maybe helped someone in thinking about these topics, and making a decision in this regard. And like and subscribe. And let me know if you're interested in another chat with Sven, potentially about neuroengineering or other problems. We could get deeper into the theory of science, philosophy of science, what is science, what is the actual guiding philosophy and drive behind it. Yeah, let me know in the comments.